Hey, this is Zagam from Z Biology. Today we are going to discuss aerobic respiration. You remember from the previous video we discussed what is respiration, its types, and the anaerobic respiration. In this video we are going to discuss aerobic respiration. Let us consider this as a cell. This is the cell membrane of a cell. Now, the first step in aerobic respiration is always glycolysis. Glycolysis is the breakdown of glucose. Now, first of all, glucose enters into the cell with the help of a transporter called as GLUT transporter. Firstly, glucose is converted into glucose 6-phosphate. The phosphate here is donated by ATP. The enzyme used in this reaction is hexokinase. Then glucose 6-phosphate is converted into fructose 6-phosphate by the enzyme phosphoglucose isomerase. Next, fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Here, the phosphate again is donated by ATP and the enzyme used is phosphofructokinase. Fructose 1,6-bisphosphate then splits into two equal halves. Two 3-carbon intermediates are formed, which are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Both are isomers of each other. The splitting reaction is done by the enzyme aldolase. For the next step, the dihydroxyacetone phosphate is converted into its isomer glyceraldehyde 3-phosphate. That means we have now two molecules of glyceraldehyde 3-phosphate. The enzyme used here is triose phosphate isomerase. Now that we have two glyceraldehyde 3-phosphate, that converts into 1,3-bisphosphoglycerate. Here, an inorganic phosphate is provided to glyceraldehyde 3-phosphate and NADH is yielded. The enzyme used here is glyceraldehyde 3-phosphate dehydrogenase. Next, 1,3-bisphosphoglycerate loses a phosphate to ATP and 3-phosphoglycerate is formed. The enzyme used in this reaction is phosphoglycerate kinase. Both these reactions are reversible reactions. 3-phosphoglycerate then is mutated. The phosphate group is moved to carbon 2 and the result is 2-phosphoglycerate. The enzyme used here is phosphoglycerate mutase. The reaction is also reversible. 2-phosphoglycerate then forms phosphoenol pyruvate. This is a dehydration reaction. The enzyme used here is enolase. Now the last step of glycolysis is the conversion of phosphoenol pyruvate to pyruvate. Another molecule of ATP is formed. The last enzyme here is pyruvate kinase. This concludes glycolysis. Now remember, all these intermediates are doubled because we had two glyceraldehyde 3-phosphate and further on each intermediate is 2 in number. For simplicity, I have just written 1. Next step is the link reaction that happens inside a mitochondria. Now here, let me draw a mitochondria first. A mitochondria has two membranes. Inner membrane has a lot of transporters. This is the outer membrane, while this is the inner membrane. Inside there is a fluid called as mitochondrial matrix. Now the pyruvate is transported inside the mitochondria with the help of this transporter here. The transporter is called as pyruvate transporter. So, in the link reaction, which is the oxidation of pyruvate, the pyruvates enter into the mitochondria and is converted into acetyl-CoA. Now, acetyl-CoA is a 2-carbon compound. In this step, carbon dioxide is released and a CoA molecule is utilized. We also gain energy in the form of NADH. Now, this concludes the second step. Now, acetyl-CoA will enter into the Kreb cycle. The Kreb cycle starts from oxaloacetate. Oxaloacetate is a 4-carbon compound, and combining with acetyl-CoA it forms citrate, which is a 6-carbon compound. Now, this was the first step of Kreb cycle. The step is catalyzed by the enzyme called as citrate synthase. Citrate is then converted into isocitrate. This is done by a series of reactions. The conversion of citrate into isocitrate has an intermediate in between, which is aconitate. These series of steps are catalyzed by the enzyme aconitase. Now, isocitrate converts into alpha ketoglutarate. Now remember, alpha ketoglutarate is not a 6-carbon compound. While this reaction is reversible and energy is produced in the cell, 1 carbon dioxide is released. So, alpha ketoglutarate is 5-carbon compound. The enzyme used here is isocitrate dehydrogenase. The next step is the conversion of alpha ketoglutarate into succinyl-CoA. Succinyl-CoA is a 4-carbon compound. That means, once again, a carbon dioxide molecule will be released and energy will be yielded in the form of NADH. The enzyme used here is alpha ketoglutarate dehydrogenase. Now, the succinyl-CoA is then converted into succinate with the release of energy in the form of GTP, which is similar to ATP. The enzyme used here is synthetase. We can say it as succinate synthase. Succinate is then converted into fumarate with the help of an enzyme called as succinate dehydrogenase. Now, this enzyme is present in the inner membrane of mitochondria. A molecule of FADH2 is released here. The next step is the conversion of fumarate into malate just by an addition of water molecule. The enzyme here is fumarase, and lastly malate is converted into oxaloacetate. We also get energy in this step in the form of NADH, and the enzyme used here is malate dehydrogenase. Now, this concludes the Kreb cycle. Let us just quickly tally up the number of ATPs, NADH and FADH2 we got from the whole step. This is the previous chart. We have 3 steps: glycolysis, oxidation of pyruvate and the Kreb cycle. And we get 3 energy intermediates: NADH, FADH2 and ATP. In the whole pathway, we have this only reaction where NADH is obtained. Now, because glyceraldehyde 3-phosphate are 2 and they form 2,1,3-bisphosphoglycerate, so there are 2 NADH released in this step. These 2 ATPs are used, so we will only consider these 2. So we get a total of 4 ATPs. No FADH2 is produced in the process. Now, for the second step, the link reaction. Everything inside the mitochondrial matrix will be doubled because 2 pyruvates enter into the mitochondria instead of 1. So we get 2 NADH, no ATP and FADH2 in this step. Krebs cycle has yielded a total of 6 NADH, 2 FADH2 and 2 ATP in the form of GTP, of course. So for the tally up, this makes a total of 10 NADH, 2 FADH2 and a total of 6 ATPs, which are then combined to form 38 ATPs in the end. So one molecule of glucose yields 34 to 38 ATPs. Hope it all makes sense. Please remember to like, subscribe and comment. Give me your questions in the comment section below.